this, and I'm going to mute everybody. Dennis, make sure we can get you unmuted. There, I got you unmuted. We had a conversation earlier. If in the middle of this you feel you absolutely compelled to ask a question, you can unmute yourself or you can use the chat, just say that time. Otherwise, Dennis will probably take a break in the middle. So Dennis Drayna is our speaker today. Dennis is a new Oakmont resident. Dennis, what, about a year now?
Yeah.
Yeah. And he comes to us care of the NIH and a life spent in this crazy world of genomes and what they do. And if anybody missed it, he gave a nice talk at the Sunday Symposium a few weeks ago, which is on the symposium website if you want to pick it up. But Dennis is here to tell us a little more about the whole area of CRISPR technology. So Dennis, you want to take over, and you're on.
Okay. Let's see. So I'm assuming everyone can now see my screen. Yes, I hope.
Correct.
Okay. Well, it's great to be here. I want to thank George and the Futures Club for the actually for the idea to come and give a talk, and for the invitation. So it's always fun to talk about great and powerful new things in the world of genetics. So let's see, here we go. Introduction and overview. So CRISPR-Cas is a system for modifying DNA, initially discovered in bacteria and subsequently shown to be useful for modifying the DNA of almost any organism. Second main point is CRISPR-Cas methods are evolving rapidly, and people are thinking of ways they could be employed to improve human health. Third main point is that CRISPR-Cas applications in humans are currently limited by ethical and regulatory concern, technical, ethical, and regulatory concerns. And then I'm going to finish by talking about some potential applications in people. Okay, so first, the CRISPR-Cas system. So CRISPR is an acronym that stands for clustered, regularly interspersed short palindromic repeats. So what are these? These are sequences of DNA found actually initially in the genome of bacteria, in which they're sort of, they occurred clustered. They tend to be sort of near each other. They have regular interspacing within that region of the bacterial genome. And they're quite short. They're often, I don't know, 20 to 50 letters, DNA nucleotide, which represent the letters of the genetic code, 20 to 50 DNA nucleotides long, and they're typically palindromic. That is, they are, due to the way they were put into the bacterial genome initially by the bacteria, they're palindromic. The sequence reads the same forward or backwards, okay? And then there's Cas, which is the so-called CRISPR-associated system. This is a protein enzyme that actually does the cleaving of the DNA that I'll talk more about in a second. The system was discovered in bacteria, actually, and here's sort of the history. Viruses, like all living organisms, bacteria, like all living organisms, are susceptible to viruses. They're called bacteriophages, and they are ubiquitous. They're everywhere. And when these viruses attack the bacteria, they usually adhere to the outside and inject their DNA in, and the viral DNA, the bacteriophage DNA, commandeers the metabolic machinery of the bacterial host, just like all viruses do, and uses it to specify creation of huge numbers of itself, and then lysing open the bacteria to liberate them into the environment, thereby completing the virus life cycle. Bacteria have, not surprisingly, developed ways to fight these things. And the way it works is when the viral DNA gets injected into the bacteria, the bacteria can recognize that it's not its own, that it's something foreign. And so what this system does is it takes little pieces of that virus DNA and incorporates them into its own genome, okay? And then those sequences, which originate, little bits of sequence originating in an attacking virus, are passed on to all of its offspring, all of the offspring of the bacteria, okay? So when that bacterium or its offspring experience infection by that virus again, the CRISPR-Cas system is able to attack the invading DNA and chop it up in places with the Cas enzyme that were previously recognized as foreign and incorporated into its own DNA. You can think of it as a vaccination. So this is a way bacteria develop immunity to defend themselves against viral attack, okay? The system was discovered actually just down the road in Berkeley by Jennifer Doudna at UC Berkeley, and sort of co-discovered by Emmanuel Charpentier, who's at the Max Planck Institute in Germany. And at a very early scientific meeting on these things, they realized they were working on the same thing. And they said, well, rather than compete, maybe we should just work together. And so they did, and have published almost all of it, co-published it together. And they were awarded the Nobel Prize in chemistry, interestingly enough, in chemistry this last fall. So, and the reason for that is because the system, even though it was discovered not that long ago, only a few years ago, discovered to be useful for genetic modification, it's been exploited enormously across many, many, many fields of biology and many, many areas of research. And it's clear that it's already made a very major impact on basic biomedical research. So that's why it was awarded the Nobel Prize. Okay, so here is the system. Now, forgive me here. I'm trying to figure out a way to minimize this. Okay, this is how CRISPR-Cas works. So the Cas9 enzyme is a protein, okay, that is illustrated in the, oops, sorry, everybody, illustrated in this figure here as this white cloud, okay, this white blob-like structure. And the CRISPR system relies on something called a guide RNA, this thing right here, this thing here. It originally, in the initial discovery, it was actually a pair of RNAs, but in modern technology development, these things are now incorporated as so-called a single guide RNA. And this RNA surveys the genome of whatever it is that's available to it. So if this guide RNA is inside a human cell, it is surveying the entire human genome up and down and up and down until it finds a match, okay, where the genetic code of the guide RNA is the complementary match of the target region in the human genome, okay? Then the Cas9 enzyme recognizes this and it makes a cut, all right? And if you want, so this cut can just simply inactivate the DNA, it will break the DNA, which is basically what the bacteria does to the virus using this system, and renders the attacking viral DNA dead, okay? And it can do the same in another organism, or you can add your own program DNA, and it turns out the cells of all organisms, but higher organisms like mammals in particular, are very, very good at finding these sorts of breaks because they are not good to have in our genome, and it will repair them. And in fact, it can repair them lots of different ways. It can sometimes seal them back up just as they were before, resulting in no net effect on the host DNA. But a lot of times it will repair them not quite right and put a mutation in there. And in fact, if you give it something, if you like make a DNA-based suggestion, it can actually, using something called mismatch repair, it can actually copy that in to this broken site, and you can add your own little sequence into this area, okay? All right, so here we go. CRISPR-Cas can be adapted to engineer a cut in any DNA. The guide RNA recognizes the DNA sequence, and the Cas enzyme cuts there. I should tell you, for using the system in higher organisms, humans, other higher organisms, we don't have a Cas enzyme. So you have to either get the gene encoding the Cas enzyme, or you can actually inject the protein itself along with your single guide RNA to actually make this all work. So the cut can be left to be repaired, sometimes improperly by the cell, resulting in a loss of gene function, or exogenous DNA can be added to direct the cell's repair machinery to incorporate a sequence of one's choosing. So I should mention, I think the most remarkable thing, I think, that surprised people about this was that it can work on DNA of an organism as large and as complex as a mammal. Bacteria have very small genomes, and bacterial viruses have even much smaller genomes. But in fact, they an enormous genome. So a typical bacterial genome might be maybe 3 million base pairs in length. Well, humans have a genome 1,000 times that. Our genome is 3 billion base pairs in length. And this guide RNA can actually survey those vast stretches and find the one place where it is a proper match. The other thing about it is that, of course, unlike bacterial DNA or bacterial virus DNA, which is more or less naked, human DNA is so long that in order to be efficiently managed by the cell, it spends much of its life coiled up into structures condensed, massively condensed and coiled up with proteins into structures that called chromosomes. And even when it's complexed with all those proteins into chromosomes, the Cas RNA can survey and find its site, or the guide RNA, and the Cas enzyme can cut at that site. And so, in fact, it actually works in higher organisms. Okay. Now, some technical advances. I mentioned that now, if you want to do your own engineering, you used to have to figure out how to get 2 RNAs in there because there's a so-called a protospacer. Let me go back and just show you. There's this little yellow here. So there's a required spacing gap adjacent to your guide RNA that the system needs to be able to see. Okay, that's recognized with a second RNA that is now combined together. So you only need to make 1 RNA, and they always go in together. Another area of technological advances are new Cas enzymes. So the classic is Cas9, but a lot of people like Cas12. But there's a whole bunch of Cas enzymes that are now known, and some of the good ones raise the specificity, and they remove the requirement for the so-called NGG palindrome associated spacer. But in any case, this little spacer here has very specific space requirements. Originally, you could design a guide RNA, but if it didn't have a GG nucleotide with at least 1 base pair in front of it, an NGG, could be anything, but it had to have an NGG, then the whole system wouldn't work. Okay, well, needless to say, there are lots of places you'd like to cut in the human genome where there's no GG in the right place. So one of the advantages of some of the other Cas enzymes is that they have removed the requirement for that and opened them up to many more broad applications in modifying the genomes of higher organisms like mammals. Okay.
Dennis, can I ask a quick question?
Yeah, sure, question.
Does that mean that sort of at this point, there's a solid group of researchers that can say, we have a reason to want another Cas enzyme, and here's how you design it?
Well, there are many reasons to want more Cas enzymes. I think everybody agrees on that. The question is, how do you get them? And the strategy to date has been to just look through more and more bacteria, more and more different strains of bacteria to find in their genome. It doesn't take long to sequence the genome of a bacteria these days. And you just look through that genome until you find something that is obviously a Cas gene, which can be done by sequence similarity to the known Cas genes. And you express that one and see if it has the characteristics that you're looking for.
Got it.
Okay. Now, people have talked about actually modifying sort of directed mutagenesis of Cas enzyme genes to try and engineer desired properties into them. And there are people starting to do that now.
Okay.
Okay.
Yep.
Now, the other thing is the guide RNA, this thing here, where's my cursor? This is an RNA, okay? But if you want to fill something in here, you're asking to make DNA, all right? And normally in the classic paradigm of molecular biology, DNA codes for RNA and the RNA codes for proteins, okay? That's the sort of the standard paradigm of molecular biology. Well, it turns out there's a way to take information in RNA and turn it back into DNA. And that's through an enzyme called reverse transcriptase. And a number of, for instance, these enzymes were discovered in viruses that have RNA as their genome. And they're so-called retroviruses because they run the normal process backwards. They go from RNA to DNA. And that copy of DNA can get inserted in your genome. So now, the most famous of those kind of viruses is HIV, the virus that causes AIDS. All right, and that's why it's sort of impossible to actually cure AIDS, because the AIDS virus has made a copy of itself and has inserted it in places in your own DNA. And your body just happily copies it and transmits it, you know, as cells divide. And so it's tough. In any case, I'll just add a personal note here. Reverse transcriptase was discovered around 1970 by Howard Temin at the University of Wisconsin, the guy who ran the first lab I ever worked in. I was the solution maker. I didn't start at the bottom of the scientific career ladder. I did not start as glassware washer. I came in at a high level, solution maker. In any case, I'd been working in Howard's lab for, I don't know, about 7 months, and he won the Nobel Prize. And, you know, I was a kid from a small town in Wisconsin, and I thought, this is great. Anyway, so reverse transcriptase drives the synthesis of an exact DNA copy of the single guide RNA. And that allows more control over the resulting genetic modification at that site. In other words, you're no longer asking the cell to just repair the cut at that site. You're actually, you've actually engineered the reverse transcriptase associated with this system to actually allow you to make an exact copy of what you want. So that raised the efficiency of the whole business substantially. Okay, so what are some current difficulties with this technology? Well, the first one is target site specificity. So-called off-target actions can now be reduced by bioinformatic tools that survey the entire genome. Okay, so we know the sequence of the human genome and the mouse genome and the rat genome and whatever, and we can look through those 3 billion base pairs to see if there's any other things we want to avoid single guide RNA sequences that target more than one place in the genome, okay? So that is great that we have these huge genomic databases that allow us to do this in, you know, 1 second of CPU time. However, many genes belong to gene families, and related family members are sometimes impossible to avoid. The biggest gene family in humans is the gene family that encodes the receptors in your nose for smell. There's about 330 functional smell receptor genes in your genome, and a lot of them are very much like each other. So they're so much like each other that you could probably never develop a CRISPR-Cas targeting that would uniquely target any one of them, okay? But, you know, that's kind of, when it comes to health concerns, that's kind of a trivial hurdle, but lots and lots of targets for lots and lots of other health conditions, in fact, are mediated by the products of genes that come in gene families. So that is going to remain really an unavoidable issue. Second thing is, of course, what Cas9 does is the first thing it does is it breaks the DNA. It makes a double-stranded cut. And chromosomal DNA in higher organisms they don't like this. DNA, they have very active mechanisms to try and repair such breaks, but they, as I alluded to, they can't always repair them perfectly. So sometimes these breaks, they'll just take the broken chromosome arm and they'll ligate it, they'll hook it onto some other chromosome arm. Well, that leads to a chromosomal abnormality, and chromosomal abnormalities are basically what cause cancer, okay? So that's a very serious concern, but people are working on making the entire process, the cleavage and the repair much more efficient, so that naked broken chromosome end isn't hanging around much. That is hopefully reducing this problem. Okay, so I didn't previously talk about this, but I think it's important to put CRISPR-Cas into context. There are many technologies to change the genes of organisms, okay? And a lot of them have been the subject of Nobel Prizes. They're in wide use for decades by the biomedical research community. And so what made CRISPR-Cas so special? I'll just make a quick run through these. The first is transgenic animals. You just inject a gene into an oocyte, an egg, and it hopefully goes into the genome intact. There's little or no control over where it actually integrates into the genome, and thus how that gene is regulated. But nonetheless, it's used and it's real easy to do. And Tim Stewart, who was at Harvard, but subsequently a colleague of mine at Genentech, was the guy who invented this and made the so-called Onco mouse, which was the first patented animal strain based on genetic modification of a whole organism's genome. The Patent and Trade Office allowed that patent. Okay, then there are methods for making targeted mutations in an animal's genome. And this was, Nobel Prize was awarded to Oliver Smithies and to Mario Capecchi for figuring this out. These methods allow you to knock out a gene. You can accurately and specifically destroy any one gene in a mouse, for instance, and therefore make a mutant. You want to know what that gene does. Well, you knock out that one gene and see what's wrong with your mouse, okay? You can also use this to make knock-ins, that you can take a specific, usually human gene mutation and engineer that mutation into the equivalent gene in a mouse. So you've sort of made a mouse model of a human genetic disease, all right? And this is a very major technology, many Nobel Prizes. The problem is you need something known as ES cells, embryonal stem cell lines. You need to do this, modify the genes in a cell that you can implant into a mouse, a pseudopregnant female mouse, and that cell will ultimately grow into an embryo and be born as a mouse, okay? An entire organism. And very few types of cells, stable cell lines that you can grow and maintain in a dish, have that capability, okay? And in fact, there are those kinds of cell lines available for only a few organisms. There are mouse ES cells and there are rat ES cells, but there are no ES cells for many, many other organisms, okay? Then finally, I want to talk about gene therapy. So the idea here is to engineer a normal version of a human gene into a small, harmless virus, okay? The one that's popular now is a little virus, tiny little virus, can only really hold one gene, usually, called adeno-associated virus. And then you can infect humans. This virus is often injected into a particular organ or site of interest. You can infect humans that carry a mutation that renders that gene non-functional. That is, these humans have some sort of genetic disease. And you can put the normal, so that they have a genetic disease because their version of that gene has a mutation in it, okay? What you can do is you can put the normal version of that gene into a little virus, and you can inject that virus into those humans, and that virus will go into those cells and make the normal gene product, and in some cases, correct the genetic disease, okay? But it is at the moment very specialized and very organ specific, all right? It also had a very unfortunate history because one of the first times they tried it at Johns Hopkins, a fellow young man by the name of Jesse Gelsinger, developed, it didn't have an extremely serious genetic disease, and he was otherwise very healthy. And they injected him, and the disease, the virus took over, and he ultimately died. So given the fact that he was more or less healthy, he had a genetic disease with a relapsing and remitting course. So they waited until it had remitted and he was feeling great, and then they tried to treat him and it killed him. This set gene therapy back at least a decade. So the problem with gene therapy is we still have limited control over how much of the gene product is made. So it can only be used in cases where the body can tolerate a wide variation in how much of the stuff you make. Okay. All right. It can't be one of those things where you have to have exactly the right amount, not too much, not too little, in order to correct the genetic disease. All right. Questions?
Okay. And from here, Dennis, you're headed into
Applications.
applications. Okay.
Okay.
Okay, let's go on.
Everyone must be in stunned silence.
I have a question.
Sure.
Oh,
Okay, so it sounds to me that this is not anywhere near being able to be used to correct for gene therapy. But the possibilities of 20 to 30 years down the line, is that possible?
Well, both CRISPR-Cas and gene therapy actually are being used today, have been used successfully in humans in extremely specific and limited instances. Okay.
Right.
So the technology is here and it's working. The problem is you need these very special circumstances because at the moment these technologies still have so many limiting constraints. So you have to find an application where you're free of those constraints. But when you can, it works. Gene therapy is now routinely used. Inject the virus into the eye. I know, don't think about it too hard. To correct a fairly common cause of a genetic blindness called Leber's hereditary optic neuropathy. There was no possible imagining a cure for this kind of a disease, and they took the normal version of the gene and they put it in adeno-associated virus, and they inject it into the eye, and it works. These people get their vision back, and it's a miracle. But it's under very specific and limited circumstances, and Leber's optic hereditary neuropathy is not all that common a disease.
Right, right.
Okay.
Well, the reason I ask is because I have an inherited genetic problem that I'm getting ready to do some whole exome testing to see if we can actually find out what it is I have in common with my mother. And then I'm sitting there thinking, this obviously is not going to be for me, but it's like for my children. When they come down, this is a late, it manifests itself in your 60s. My children are in their 30s and 40s. And is there hope for them down the line with CRISPR that this might be something that they could actually avoid the ataxia?
Lee, let me step in here and get Dennis to finish up, because Dennis is about to talk about what CRISPR may be able to do and where the edges are.
Okay.
Yes, I would say just two things. I would say there is hope. Maybe not with CRISPR, but maybe with something else. But I should also say that when it comes to predicting when a technology, a human health scientific discovery will become applicable, I have learned that I inevitably estimate longer than it ends up taking.
Yeah, I understand that.
Okay, so applying CRISPR, I think, let me just talk about several general topics here. First of all, research tools. So what CRISPR is used for now is to make research tools, okay? That's why it's been adopted by thousands of biomedical researchers working on all sorts of things in all sorts of organisms, is to make tools for research. And that's really what got it the Nobel Prize. It is not it is just beginning to find specific applications in which it can be successfully used in humans. But the vast majority of its uses have been to make research tools. I'll tell you, the people who are now sort of on this business are the livestock improvement people. Livestock improvement is thought to be a place where lots of things could be done very quickly with CRISPR that avoid many of the sort of ethical and regulatory problems of working in humans first. Okay, so the second thing is the problem, which cells do we modify with CRISPR? And that addresses the issue of germline versus somatic changes. So let me explain that a little bit. CRISPR modifications to most of the cells of the body will not be passed down to offspring, okay? The one aspect of where, well, actually there have been two aspects, one of which it was applied to human oocytes and which were implanted, fertilized and implanted, and gave rise to the birth of a total of 3 children in China. And this was illegal, and the guy who did it is currently sitting in jail. It was an attempt to make the father in this case was HIV positive, and it was an attempt to alter the genes of his offspring so that they would be resistant to HIV. Another area which has been successfully done is people have CRISPR-Cas modified bone marrow cells. So this is a type of stem cell, and you remove the bone marrow. They treated those cells, the ones that give rise to red blood cells. So there are lots of different kinds of cells in your bone marrow, and one kind of cells gives rise to your red blood cells as opposed to your white blood cells. And those, then the bone marrow of the subject was completely ablated, completely knocked out with toxic agents, and their own now modified bone marrow was reinfused, and it cured them. Okay, this was for sickle cell and also done for blood disease called thalassemia. So those just reside, they will grow and divide and live on, but only in the bone marrow of those individuals. They don't affect the gamete producing cells, the eggs and the sperm of those people, okay? CRISPR modifications to germ cells, such as occurs when CRISPR is applied to an egg that is subsequently fertilized and implanted, because that gives rise to all the cells in the body, including your germ cells, will cause a modification to be passed to all future generations, okay? So this is J.K. Hay and his genetic modification of whole humans. In China, J.K. is now sitting in jail for having done this, but the real issue is, in addition to off-target effects, and we don't know what other health impacts, long-term health impacts could happen to people who've been CRISPR'd as they grow up and get older and develop. But there's this issue of, do you want to start changing things for future generations? They, are not around to vote on this. So, so what do we wish to change? So one thing that people talk about now are somatic mutations in cancer. So what is cancer? Most tumors are not caused by inherited mutations. They're caused by mutations that are acquired in various cells of the body, like a mutation caused by cigarette smoke in the cells of your lungs, okay? Tumors are the result of acquired mutations in various somatic tissues, resulting in a line of cells that lose control of their own growth, okay? So could we find and then correct those mutations in tumors using CRISPR? Well, a lot of tumors, for instance, are now known to either to have mutations in one gene called P53. I didn't put that down here, but P53, there are P53 negative, over-expressing tumors where P53 is too active, and you might want to just go into those tumors somehow, just the tumor, using CRISPR-Cas, and kill P53 using CRISPR-Cas, which I think is something people think that could be done. Other tumors have a P53 insufficiency, so perhaps you could use CRISPR-Cas to turn up their existing P53 and thereby bring growth control back to these cells and thereby basically cure the tumor. So these are somatic mutations, and those as a target for CRISPR-Cas people are beginning to talk about. Another thing people are beginning to talk about is whole body germline mutations that cause genetic disease. So lots of genetic diseases, things like sickle cell disease or cystic fibrosis, or a gene I found, hemochromatosis. That hemochromatosis is the most common disease gene in Europeans. 1 in 11 people is a carrier. It's an iron overload disorder, and people still die of it, unfortunately. These genes, the mutations that cause these are passed down from generation to generation. In fact, there are 400 different mutations in the cystic fibrosis gene, but over 70% of them are a single mutation, so-called delta F508, a deletion of phenylalanine at amino acid position 508 in the CF protein. Okay, hemochromatosis, not only is almost all of this disease one gene, but 92% of it is one mutation in that gene. So one could go after these mutations with CRISPR-Cas, but the problem here, of course, is that if you did whole body germline mutations, so if you did injections into an embryo or something like that, then these would be passed down. And this gets us into the field of eugenics, which is the genetic improvement of humans as a species. And eugenics has first a very sort of a good reputation. In the 1920s, it was developed, sort of the thought leaders were all Americans. And then the ideas were adopted by Nazi Germany in the 30s and led to obviously enormous problems. And so eugenics fell into complete disrepute. And now people are sort of almost talking about a reemergence of eugenics as a way to fix some of these, because these gene mutations, cystic fibrosis mutations and sickle cell disease, 1 in 24 people carries a cystic fibrosis mutation. There are many millions of people walking around the population that have these mutations, okay? So maybe we could just get rid of them, sort of clean up the genome of the humans. Then there's this issue of tissue-specific germline mutations. So rare mutations that are present in all cells of the body, but only cause disease in one organ or one tissue. So classic here is deafness. Okay, there are about 150 different deafness genes, and those mutations are in every cell in the body, but the vast majority of those don't cause anything other than deafness. So for some reason, the inner ear is uniquely susceptible to the problems caused by that mutation. And so the people are just deaf and nothing else. Muscular dystrophy is another one. Polycystic kidney disease, it's the single largest cause of dialysis, PKD, polycystic kidney disease. So all of these are inherited. They're rare, relatively rare mutations, but there are ways that they are just expressed in a single organ or a single tissue, where one could imagine engineering a CRISPR-Cas vector and a CRISPR-Cas system and going after them to just try and change them. And finally, another one I think has been discussed a lot is a true cure for AIDS. I mentioned the reason AIDS is uncurable is because there's a DNA copy of the virus sitting in your own genome and it ain't going anywhere. Well, CRISPR-Cas could easily find that copy in the genome of a human and cut it and inactivate it. So it could ultimately produce a true cure, a genuine, permanent cure for AIDS, which I think a lot of people are excited about. So, so what about common diseases? Okay, I've talked to you about lots of things, sickle cell disease or cystic fibrosis. These are rare, relatively rare genetic diseases. What about common diseases? Well, common diseases are common because they're the sum of many different diseases. So cancer is at least 50 different diseases. Any oncologist will tell you that kidney cancer is extremely different than brain cancer. And another point is that almost all diseases have some genetic contribution, almost all. Okay, there's some what's called heritability, some contribution of genes. However, the original or the individual genetic variants that underlie common diseases are coming to be known, but almost all of them exert very small effects on the development of the disease. They're not like cystic fibrosis or hemophilia or sickle cell disease, whereas if you have the mutation, you get the disease, right? Most of the contributors to complex diseases like heart disease or metabolic disease or psychiatric disease are clearly contributors. The evidence that they play some sort of role is overwhelming. However, the effect sizes are very small. So having or not having any one of them is not all that predictive and not all that diagnostic and not really at all prognostic for the subsequent development of disease. However, they're finding lots of them. They're finding lots of genetic variants in lots of genes that each make a small contribution to the development of schizophrenia or type 2 diabetes or you name it, right? And these genetic effects are being added together to produce now what are called polygenic risk scores that do seek to be more predictive of the effect of this sum of small genetic differences on your development of disease, okay? I would say at the moment, CRISPR is only worth considering for changing human genetic variants that have large and predictable effects. They're good for something like hemophilia. You know, there's only one gene in which mutations cause hemophilia. And if you have a mutation in that gene, you're gonna have that disease. Well, CRISPR-Cas could be unleashed on that sort of a thing. These common diseases where there's lots and lots of little contributors, I think for the moment it's safe to say are probably out of reach for CRISPR-Cas. They just are. Now, I'll just say one last thing. I'm sort of I don't know, could be politely described as a crank in human genetics. And I am a believer that common diseases, in fact, are not all these kind of variants that each exert a very small little effect, that there are rare variants with very large effect that are important in many common diseases. And it's just that the genetic studies that have been used to find these variants, they're just invisible to that study design, okay? And you have to do whole genome sequencing on thousands and thousands of people to find rare variants of large effect. But when you find rare variants of large effect, well, then that's something to think about for CRISPR-Cas. Okay, so summary and thoughts. CRISPR-Cas is a powerful new genetic technology that is starting to be applied to humans. All uses of CRISPR in humans will be considered experimental and thus highly regulated and, importantly, not subject to insurance coverage for the immediate future. You will not, anybody who's talking about CRISPR-Cas to treat some human disease, insurance isn't going to cover anything, okay? Technical issues such as suitability of target sites and off-target effects will continue to limit the technology, in some cases, I think perhaps forever. And finally, sort of a note of general optimism, many of the ethical issues concerning safety and germline transmission have previously been dealt with for traditional gene therapy. So a lot of these arguments have been made and hashed out among bioethicists and institutional review boards who actually oversee the safety of human subjects research. So some of the path has already been paved for some of the ethical implications for using CRISPR-Cas in humans. So that, I think, is my last slide, and I'm glad, thank you very much. Glad to have given you this sort of breathless dash through CRISPR-Cas, and I'd be glad to take any questions.
Okay, so let's open up. Let me start with a fairly obvious one. In the cases where we have had an attempt to use CRISPR in humans, okay, is there any evidence of a near-term side effect problem at all?
Not yet.
Okay. So.
But I should tell you, the experiment that was done in China was done to disable a gene called CCR5, which is a naturally occurring mutation in CCR5. Is not that rare in the population. About 10% of most people outside of Africa have this mutation. And so you have to have both copies of the mutation to make yourself resistant to AIDS. So 10%, the probability of any one person getting it from their mom is 10%, multiplied the probability of getting it from their dad, which is 10%. So 1 in 100 people has 2 copies of the CCR5 mutation, and they're resistant to AIDS, just naturally, okay? CCR5 codes for the receptor that the AIDS latches onto to get into the cell. That's a unique gene. It's not a member of a gene family. So off-target effects actually for CCR5 are not such a worry. Yeah.
Okay. Okay, other questions at this point?
Was CCR5 the one that the Chinese geneticist changed in the children?
Correct.
And yet it does occur naturally and in people, and yet he got in trouble. I'm a little confused about that.
Right. Right, he got in trouble for a series of rich and complex reasons. So first of all, he did it in secret. He never told anybody he was doing this. Second of all, he did it not in the course. So J.K. Hay worked in a department of reproductive medicine. Okay, so he was used to in vitro fertilization and embryo implantation in humans, a regular part of his business in a department of obstetrics. But all this was actually done in a private company that he was part owner of, that he didn't tell anybody about. So that was another little problem. So this was done in a for-profit setting with no regulatory oversight. And since he didn't tell anybody, there was no general evaluation of the larger risk by an independent group of experts who might have had maybe a little more information about this than he did. Yes, it is true. I mean, he felt that the intellectual case for doing it was compelling, but this is permanent, and the change that he made. So I should fess up here. I do not recall the exact change he used CRISPR-Cas to make in the 3 kids. Okay, I think.
So it may not have been exactly the same thing that.
I think he tried. He tried, but CRISPR-Cas is oftentimes not as, especially at the time he was doing this, not as perfectly accurate and as reliable as one might want. And so it was always possible that it put some other mutation in there that could have had who knows what kind of effects, because CCR5 does not exist on our cells to provide an entryway for AIDS. It has a completely different function that we all need. If the CRISPR-Cas modified it in some unexpected way, which there was a reasonable chance of happening at the time he did this, then the kids and all of their offspring and so on could have been left with a significant problem.
Okay, I understand what you're saying. It wasn't as clear as, well, this is naturally occurring anyway. There was a lot more in between there.
That's correct. In the one that was done to cure sickle cell disease, that one was very clear. They didn't just cure the sickle cell mutation. What they did was they left the sickle cell mutation intact in that patient. They did a clever trick. It turns out there are 5 different hemoglobin genes, including one that is turned on only in fetal development. And there's a hereditary condition called hereditary persistence of fetal hemoglobin, where people don't have adult hemoglobin, and it doesn't seem to hurt them. They go live a normal life, but fetal hemoglobin doesn't sickle. Okay, so what they did was they found a place in the genome that normally turns off fetal hemoglobin production at birth, and they use CRISPR-Cas to knock it out. Okay, and it doesn't seem to regulate anything else other than the fetal hemoglobin gene. So they knocked that out, and sure enough, the repression of fetal hemoglobin was relieved. Fetal hemoglobin turned on in what was now an adult patient, and their symptoms of sickle cell disease stopped. Okay, so sometimes you can get something specific enough that now, of course, that's not germline transmission, but I think the clinical outcome in that particular case was so good that people are thinking, well, maybe we should think about trying this in a germline situation.
Dennis, if you look at the major research hospitals in the Bay Area, okay, Stanford, UCSF, your pick. If a valid researcher associated with the genetic world, so they were knowledgeable, came up with a proposal to do a CRISPR modification in a human, so that would be a traditional research proposal within the institution, would that typically now need to go at a higher level than the institution to be tried?
Technically, it would not. Technically, that regulation is done at the institutional level.
Right.
But I think, given the electric third rail of CRISPR-Cas, it would almost certainly go to a higher level. And in fact, if I were a dean at UCSF, I would probably insist on review at a higher level, just to make sure my institution was fully covered from top to bottom.
If one of the genetic research programs in the Bay Area were to actually do something novel in humans, which one would it be?
Oh, good question. There might be some, so I'm not in the thick of it, but there might be some under discussion now. I would say it would be equally likely at UCSF or Stanford. They're both
Okay.
that kind of place. Stanford has chosen, in terms of a medical school, in terms of a medical center, Stanford has kind of chosen to become an institution of extreme specialization. And this, of course, is obviously something that would need tremendous specialist expertise. So maybe Stanford might be a little bit more institutionally interested in it. But, UC has traditionally a very strong medical genetics department. And I wouldn't count them out in any way. UCSF. Okay.
Okay. Want to take you to messenger RNA vaccines for a moment.
Sure.
Okay. Those do not have any effect known on genes.
That's correct. There's no reverse transcriptase in humans or other higher organisms. Humans have ways of, anyway, they have a mechanism for generating genetic diversity called retrotransposons that go through an RNA intermediate that then gets turned into DNA, but there's no reverse, humans do not have a reverse transcriptase. So yeah, it's
Okay. Since a lot of us are getting this magic new vaccine, there's a moment's worth of thinking about that. Okay, any
right.
other questions for Dennis at the moment?
Yes.
I would just, as a former virus guy, I would just interject that RNA vaccines are, they're they're probably extremely safe. You know, the reason a lot of vaccines have problems or adverse reactions is because of the junk that comes along with them when they're made. So for instance, the flu vaccine is grown, influenza is grown in eggs. And if you're allergic to eggs, you can't get the flu vaccine because it's got all this egg stuff that gets injected into you at the same time. So many vaccines, the problems with many vaccines cause with the junk that comes along with them. And of course, an mRNA vaccine, it doesn't have any junk. It's extremely simple. The traditional worry about mRNA vaccines is we don't make antibodies against our own proteins, right? We can't. If we do, that's called autoimmune disease, and it's really bad, right? So we all have what's called immune tolerance for all the proteins that we grew up with in our bodies, okay? So the proteins we make are hidden from immune surveillance. Well, what you're doing when you inject an mRNA into your shoulder is you're asking your muscle cells to make a new protein. And it's possible that your immune system might not be so good at recognizing this as foreign because it's made by us. And we already have mechanisms, robust mechanisms of immune tolerance for the things we make. So that, I think, was a big concern with mRNA vaccines.
Okay, I think that's going to be where we're going to end up. 2 weeks from now, Mahir is going to talk about quantum computing, taking us into a completely different world and a modern world. And Dennis, sometime, if you aren't aware of this, we used to have something called the Sonoma Development Center down in Glen Ellen.
Oh, I'm very familiar with it.
Okay, then you know the history of eugenics there.
Oh, yes.
Okay. All right. Well, thank you, everybody, and we'll see you in a couple of weeks. Thank you so much, Dennis.
Thanks, everybody.